Hello, thank you for having me here. It was 30 degrees when I left Manila yesterday, very cold. So before I make my formal report, just a brief introduction of the Philippines. The Philippines is one of the mega diversity countries in the world. We have a very high endemicity. We have around 47 percent endemic species, and this is only for most vertebrates that we have studied thus far. And we have not yet covered the invertebrates and the plants. So, and on top of that, we also have what you call the super endemics. These are the species that are found only in one particular area in the Philippines and nowhere else. And I'm sure we have some of these super endemics in some of the mega diverse countries as well. So, oh, I forgot to introduce myself. So, I'm Ian Fontanilla. I'm an associate professor. I have two affiliations, one at the University of the Philippines, Diliman Institute of Biology, and the other one is the Philippine Genome Center, and they happen to be adjacent to one another, so very perfect for us when we do our DNA barcoding work. So my key taxonomic interests are mollusks and their parasites and endemic Philippine vertebrates. I have been doing barcoding even before I realized I was doing barcoding when I started barcoding nematodes. So my focus is the Palanan Forest Dynamics plot and Mindanao Island. The Palanan Forest Dynamics plot is one of those protected areas in the Philippines that has been studied ecologically for the past 18 years. So I am from the University of the Philippines, and these are my institutions. And these two institutions have been working very closely towards DNA barcoding of Philippine endemics. So this is the update for the Philippine barcodes on bold. I would like to focus your attention, however, on plants. With plants, we have around 8,000 to 13,000 species in the Philippines, of which we only have 141 barcoded thus far. And this is because we only have one lab or one institution in the Philippines from the University of Santo Tomas who's been doing plant barcoding. And I hope that will change in the next 5 or 8 years. So we have several projects planned and ongoing. The first one is the Palanan Forest Dynamics Plot Genome Observatory, which we hope to cover the entire Palanan Forest Dynamics plot, and we will cover all vertebrates, gastropods, insects, basidiomyces, vascular plants. So basically, every eukaryote, multicellular or eukaryote that we will encounter, hopefully we could barcode. So this will be the first comprehensive inventory of all non-microbial species in a tropical ecosystem, to include at least 5 individual species, hopefully more. The Palanan Forest Dynamics plot is very ideal because it has been studied for several years. We already have ecological data. It has been visited by typhoons and other disturbances for the past several years, and we would like to know how these disturbances affect biodiversity. It doesn't seem to be affecting biodiversity because no matter what happens, the biodiversity is still there. So it's a very ideal experimental lab. And then we also have a DNA barcoding of Philippine bats. This is an ongoing project in collaboration with the Field Museum. One of the project lead, Adrian Luxon, was here last, I think in 2016 for the GTI training. And now we are conducting GTI training in the Philippines with the CBD. And then this is underway. We have focused on Philippine bats. We have around 72 bat species in the Philippines alone. And then a third project, this is also ongoing, is a wildlife forensics and DNA barcoding of Philippine biodiversity. Now, at the beginning, we always had, we have also encountered similar problems like in other countries where we had to go through the bureaucracy to get permits. Because the DNR, Department of Environment and Natural Resources, is the one that is responsible for protecting our biodiversity. And then suddenly, then one of our project leaders then had this brilliant idea: why don't we just work with them? If they are the ones who are giving us the hard time, then why don't we ask them to, because they're also responsible for protecting these specimens. So they are the ones who ask us, could you barcode this? Could you do this? Could you determine what species this is? Is this a Philippine eagle feather? Is this a protected or endangered coral, etc. So we came up with a we came up with a collaboration. They provide us the specimens, and then we barcode it for them. We barcode these specimens for them, so it works. It benefits both of us. So now we use the specimens they've collected to populate the Philippine endemic database, which we use for wildlife forensics. So our goal for this project is to barcode all Philippine endemics confiscated by the DNR wildlife enforcement officers. So one of the first that we have barcoded is the Philippine eagle. And then the next one is the foodborne helminthiasis problem in the Philippines, present status, response, and proposed intervention. Now, this is an offshoot of our eDNA project to detect Schistosoma japonicum, which is a human-borne parasite. Well, it infects other vertebrates, but it can also infect humans. And then we would like to expand our coverage to other parasites, which we could get infected via soil or water. So this will be hopefully implemented starting next year. So for the SWOT analysis, in terms of strengths, we are fortunate to have a very supportive government. They do provide us with lots of funding through the Department of Environment and Natural Resources and the Department of Science and Technology. So that is our main strength. And we also have a lot of taxonomists in the Philippines. Our problem, however, is that despite the fact that we have a growing number of DNA barcoding groups in the Philippines, we still need more warm bodies to barcode them, because if they provide us grants, if they provide us funds, you have to submit grants first. So no proposal, no money. So we have this unusual problem in the Philippines in that the budget for the Department of Science and Technology and the Department of Environment and Natural Resources for research is going down, not because of lack of support, but because there are not enough takers. So there are not enough people applying for grants. So it's an unusual problem. So what we need now is to build up our capacity. We should have more. We need more PhDs to write grant proposals. Now, our major threat is a natural one. Well, it's also a man-made one, I think. It's an ongoing biodiversity loss in the Philippines. With the Philippines being a biodiversity hotspot, we have a very high endemicity, and at the same time, many of these endemics are critically endangered because of habitat loss. So, just to give you a perspective, in the 1400s, the Philippines has an area of 300,000 square kilometers of tropical rainforest, divided into three main types, and each of this type produces its own unique microhabitat, and that's why we have a very high diversity. Now, in the late 1990s, from 300,000, the rainforests have been reduced to around 8,000. So that's a significant loss in biodiversity, significant loss in habitat, and therefore a significant loss in biodiversity. So we have a problem in that we are already losing what we haven't yet identified. We don't even know if they're already, we don't know what we have already lost. So the threat is there. Hopefully it will be, I don't know if we can reverse the habitat loss at this time because we have a problem with our population. However, there are opportunities in the Philippines. We can network with the other ASEAN countries. ASEAN stands for Association of Southeast Asian Nations, which also have similar problems and also similar opportunities. In the ASEAN, we have many countries in the ASEAN also boast of very high biodiversity. And through the ASEAN Biodiversity Center, which is headquartered at the University of the Philippines, Los Baños, we could probably network with the other ASEAN countries and maybe come up with a collective effort for this DNA barcoding. Thank you very much.